Alright, this video is part 3 of the EOC review for Unit 2. It's going to be macromolecules, and we'll talk about enzymes as well, and we will get to enzymes. Now, a few elements that are key in living things are, of course, carbon, and we've talked about carbon, and oxygen, and hydrogen, very important atoms basically in everything. A couple other ones that we will mention briefly as we go. Sulfur is very important, part of proteins. Phosphorus, your DNA and ATP, things like that need phosphorus. And nitrogen. Nitrogen is essential in proteins. It's essential in your DNA as well. In the past, people have spelled things like that or other, whatever helps you. One thing that's easy, they're all single letter atoms. Now, what is a macromolecule? Well, macro means large, so these are larger molecules, at least larger than water, and some of them are smaller, some of them are very, very big. There are 4 classes of macromolecules in living things. There are carbohydrates, lipids, proteins, and nucleic acids. And each one we're going to list a function. We're going to talk about what they're made up of. And we're going to talk about what they're kind of what they do in living systems and a few other vocabulary associated with them. So let's talk about carbs first. Carbohydrates. What is their function? They're the primary source of energy for all living things. All right. Another word for carbohydrates that you'll often see is sugars. Some popular carbohydrates are things like glucose, always makes the front page, sucrose or table sugar, fructose, galactose, some others, starch, glycogen, chitin, a couple others. Carbohydrates are always composed of 3 things: carbon, hydrogen, and oxygen. And they're normally in a readily noticeable ratio, whereas the carbon is always equal to the oxygen, and the hydrogen is equal to the carbon times 2. Now, this isn't always the case, but normally it works itself out to where carbon, hydrogen, and oxygen are in a 1:2 to 1 ratio. So for instance, glucose, the chemical formula for glucose is C6H12O6. That's just a formula you need to know. Glucose, pretty important molecule. C6H12O6. You can see that regular formula there. Now, carbs represent energy for living things, and how do they deal with that? Well, plants will actually store carbohydrates in the form of starch. And so what is starch? Well, starch is just these long chains of glucose molecules. So here's a glucose molecule. It's got 6 carbons in it. It's normally shaped like a ring, like that. And you'll have these long chains of glucose molecules. Now, each individual glucose molecule is called a mono 1 saccharide. Saccharide is just another word for sugar. And so this is the monomer of carbohydrates. What is a monomer? It's just a single unit of something. It's just one monomer. The polymer, and what are some examples of monomers? Glucose, fructose is another example. The polymers of starch of carbohydrates are called, of course, polysaccharides. What's an example of a polysaccharide? Well, starch, because there's lots of monomers. There's all these individual monomers here, right? So starch is how plants store carbohydrates. Glycogen is how animals store carbohydrates. All right. Now, not all carbohydrates are stored for energy. There are some carbohydrates that are structural in nature. That means they're used to build something. One example of that is called cellulose. All right, and cellulose is a structural carbohydrate found in plant cell walls. Another structural carbohydrate is called chitin, not chitin or chitin. I've heard of it all this year. Chitin. And chitin is found in fungal cell walls, but it's also found in exoskeletons of bugs and arthropods, things like lobsters and that sort of thing. All right, so that's it on carbs, pretty simple. Next is lipids. Lipids store energy. That is their function. How are they able to do that? Because they're made of these long chains called hydrocarbons. Well, what do you suppose is in a hydrocarbon? And at each one of these junctions is a carbon. There's hydrogens in there. Carbon and hydrogen. Now, lipids will contain oxygen and other things, but they're primarily made up of carbon and hydrogen. A lipid is made up of monomers called fatty acids. An example of a polymer in a lipid is called a triglyceride. Why is it called a triglyceride? Well, there is a molecule called a glycerol molecule, and what it does is it holds 3 fatty acid chains together, thus the tri. And this is the standard lipid found. But also there is a lipid in your body, all over your body, trillions and trillions of places in your body, called a phospholipid. And phospholipids they're kind of shaped like this, whereas they have a hydrophilic head, meaning that likes water because it's polar, and a hydrophobic tail because it doesn't like water. And these phospholipids arrange themselves usually like this. Well, where are phospholipids found? They're found in your cell membranes, which we'll talk about in a later segment. Well, lipids are made up of saturated fats and unsaturated fats. Two main kinds there: saturated, unsaturated. And saturated has the maximum amount of hydrogen, whereas unsaturated fat doesn't because of double bonded carbon. So here's an example of saturated fat. It kind of looks like this. Nice long straight chain, no double bonds. Hydrogens are sticking out here. Whereas an unsaturated fat is going to have the occasional double bond. And that actually causes that molecule to bend. All right. You can have one double bond in an unsaturated fat, and it'll be called a mono unsaturated fat, or 2 double bonds, and it will be called a poly unsaturated fat. A trans fat, which you've probably heard of, or a hydrogenated fat, is taking an unsaturated fat and adding hydrogen to it, thus making it behave like a saturated fat. Saturated fats are solid at room temperature, and unsaturated fats are liquid at room temperature. These are usually associated with animal fat, and these are normally associated with plant fat. So that's lipids. Proteins. Now, we could spend a lot of time talking about proteins, but we're not going to for this video. Proteins have lots of functions. There are 6 or 7 of them mentioned in this outline. They're structural, meaning they build things. They signal, meaning that your body uses them to signal one part of the body to do something or whatever. They act as catalysts. They act in immunity, in the regulation of body processes, so homeostasis. Some animals use them as poisons or venoms, and also in transport of things across the membrane, like a transport protein. Lots of functions. Proteins are made up of individual monomers called amino acids. I'm writing it big because, please, this is just necessary. All right. And a polymer, then, because the amino acids are joined together with a bond called a peptide bond, the polymer is called a polypeptide, which is just another name for a protein. All right. Now, real quick on proteins, they have 4 structures. We call them primary. If I write a circle like that with a 1, that's primary, secondary, tertiary, and quaternary. Primary structure of a protein is just a chain of amino acids, whereas these little circles are amino acids, and the little lines connecting them are peptide bonds. It's just a chain of amino acids, like Mardi Gras beads. Well, the secondary structure is when you start to manipulate that string of Mardi Gras beads. And when left to their own devices, they will either kind of coil on themselves and make this helix, which is called a alpha helix, or they will form sheets like this, which are called beta sheets. That's the secondary structure. Well, when you get several of these alpha helixes and beta sheets kind of sticking together, that is called a tertiary structure. And the tertiary structure, in some cases, is the completed protein. Proteins have a shape, and that shape dictates what they do. A wrench has a shape, and that shape dictates what it does. This particular wrench won't do much because it looks like it's bad. All right, and so proteins have a shape. Well, some proteins need more tertiary structures. And so a protein that has multiple tertiary structures is called quaternary. And they'll kind of come together like this, forming bonds with one another. And they do something in the body. And a good example of a quaternary protein is hemoglobin. It's how your body gets oxygen to all of its parts. The hemoglobin molecule is designed to carry iron, which will in turn readily carry oxygen. So that's how proteins are designed. Lastly, but not least, in the least, is the nucleic acid. And these are things like DNA and RNA. What are they made up of? Well, the monomer of nucleic acids are called nucleotides. And a nucleotide is made up of 3 things: a phosphate, a nitrogenous base, and a 5-carbon sugar. And the sugar is how it's named: deoxyribonucleic nucleic acid. Deoxyribose is the sugar. Ribonucleic acid. Ribose is the sugar. All right, whereas the polymer is the full-on nucleic acid, the full-on DNA. Now, what is the function of nucleic acids? Their function is to store hereditary info. Extremely important, obviously, in organism development. Now, real quick, let's talk about enzymes. Enzymes are biological catalysts. What is a catalyst? A catalyst speeds up chemical reactions. Well, how do they do that? Well, a chemical reaction has a particular energy called an energy of activation. And this energy of activation is just the energy that is required to do the reaction. Well, some reactions, whereas this is time and this is the amount of energy, some reactions will kind of spontaneously happen, and they have a very low activation energy. And what are they doing? They are releasing energy. Well, this would be the activation energy right here. Sometimes you'll see that written as like that, energy of activation. All right, and this is the amount of energy that it requires to do that. Well, what enzymes do is they actually lower that energy. So if I were to draw the same line, let's say I add an enzyme to this reaction that causes it to work better, that same reaction might occur with a lot less energy needed. All right, well, some reactions, this kind of reaction releases energy. Some kind of reactions require energy. So they actually absorb it. And so they'll start down here and they'll actually absorb some energy. Well, there's a high activation energy on this one. High activation energy. And so how can we lower that? You add an enzyme. Well, the reaction might take place like this. Now, what is that doing? That's saving the body energy. It's saving tons of energy and tons of time. So the way enzymes work, remember I said all proteins have shape? Well, most enzymes are proteins. And so this particular enzyme is shaped like this. And its shape means something. Its shape is specifically designed for a particular molecule called a substrate. So here's the enzyme. Here's the substrate. Well, in this case, there's two substrates. And this enzyme's job is to go out and find these two things and to put them together. All right, and so those substrates will fit right in this little site, and this site has a name, of course, and it's called the active site. And the way this works is picture a rubber glove and you sticking your hand down on the rubber glove. Now, it's not a perfect fit. It's called an induced fit. You actually have to kind of make the glove work around your hand, but it's definitely designed for your hand. And so there's an induced fit for these substrates in here, but once they enter into the enzyme, it will actually combine. Now, notice what happened. The enzyme is unchanged, but the substrate is now changed. And so if you had lots of substrate out here floating around, this one enzyme could do the work of combining every single one of these pairs until the work was done. All right, I'm not going to do all of that, but you get the idea. Now, you'll notice, let's just say that it's all done. All right, it's all done. Notice the enzyme is unchanged, but the substrate is all used up. It is all changed. Well, sometimes instead of putting things together, the enzyme is designed to take things apart. And it will look for things that do look like this, and it will take them apart. And then that enzyme is done doing its work when everything is taken apart. It's the same idea. Some enzymes build things, some enzymes break things, but at the end of the day, it's all about speeding up the chemical reaction in order that it might work. So when the substrate is gone, the enzymes will no longer react, but the enzyme is still there, ready to do some kind of work, waiting for that to happen. A good example of an enzyme in your body is an enzyme called catalase. And catalase will readily break down hydrogen peroxide. And so you have hydrogen peroxide in your body to fight off infection and so forth. And when you put hydrogen peroxide on your knee, and because you scraped your knee on the carport or whatever, and it's all nice and bubbly, and you're like, look, it's working. Well, what's actually happening there is an enzyme called catalase is taking this molecule, which in this case is hydrogen peroxide, and breaking it apart into two groups, water and oxygen, which is the bubbles that you see. So the bubbles doesn't mean the peroxide's working, it actually means that your catalase is working.